Hello friends, I Dr. R.D. Patil, Head Department of Zoology, Vasantranik Arts, Science and Commerce College, Chada. Dear friends, today we discuss about your S.Y.BSc Zoology Zoo 302 course paper Zoology paper second semester first, that is a biochemistry. In this course, we already discussed about the completion of the syllabus point of view. There are two units. First unit is the carbohydrate metabolism. Second unit is the lipid metabolism. In carbohydrate metabolism, dear friends, we already discussed about the uh, glycolysis, Krebs cycle, pentose phosphate pathway, gluconeogenesis, glycogen metabolism, review of electron transport chain, that is TCA cycle. Next, unit number 2 in lipid metabolism, we already discussed about the biosynthesis and beta oxidation of palmitic acid, lipogenesis and lipolysis. So these points are discussed in this unit number 2. Today we start the unit number 3, that is the protein metabolism. Dear friends, we first known about the what is main protein. We are starting for this particular main topic, that is protein metabolism. We already discussed about what is main metabolism. In protein metabolism, we detail discuss about the biosynthesis of amino acid, transamination, deamination, decarboxylation, and urea cycle. These sub points are explained in your this unit number 3, protein metabolism. Dear friends, we discuss about the first what is mean protein. Proteins are the polymers of amino acids. Each of amino acids are contains a central carbon, a hydrogen, a carboxyl group, an amino group, and a variable R groups. Major function includes acting as enzymes, receptors, transport molecules, regulatory proteins for gene expression, and so on. So this is a most important to specific point of view as your introduction point of view. Proteins are the large complex organic molecules which play diverse roles in living organism. Proteins made up of such elements as already discussed about the carbon, hydrogen, oxygen, nitrogen, sulfur and phosphorus. In introduction of protein, first as the we already proteins are the molecular machineries, building blocks and arms of a living cells. Their major and almost sole functions is enzymatic catalysis of chemical conversion and around the cells. Proteins are polymers. They are built of the amino acid that are linked into a peptide chains. The chain consists of a chemically regular backbone, or main chains from which various sides chains R1, R2, dash Rm, that is also called or projects. So this is a schematic or formula of the proteins. In simple way, protein is nothing but the building blocks of the amino acids or also chain of the amino acids. There are 20 main pieces of amino acids. Reduces their position in the protein chain is a gene encoded. The peptide bond allows for rotation of protein and therefore protein can fold or fold and orient the R group of favorable position. Protein shape is determined by the sequence of the amino acids. This is a schematic representation. Almost everything that occurs in the cell involves one or more proteins. In every components which are made by the protein provide structure, catalyze cellular reactions, and carry out of myriad of other tasks. Their central place in the cell is reflected in the fact that genetic information is ultimately expressed as a protein. So in simple way, this is the most important role in the living cell, also acts as a catalyst and genetic information to express as a particular protein groups. For each protein, there is a segment of DNA, a gene that encodes information specifying its sequence of amino acid. There are thousands of different kinds of proteins in a typical cell, each encoded by a gene and each performing a specific function. Proteins are among the most abundant biological macromolecules and are also extremely versatile in their functions. So this is the uh, most important role in the cells as your uh, intracellular or extracellular point of view. This is a amino acid pools we know about the first two in your proteins are the most abundant organic compounds and constitute a major part of the body weight, that is 10 to 12 gram. In adults, they perform a wide variety of static, structural, and dynamic enzymes, hormones, clotting factors, receptors, etc. functions. About half of the body proteins, predominantly collagen, is present in the supportive tissue, that is skeleton and connective tissue, while the other half is intracellular. Proteins are nitrogen containing macromolecule consisting of L alpha synthesis alpha amino acids as the repeating units of the 20 amino acids found in proteins. Half can be synthesized by the body for non-essential, while the rest have to be provided in the diet. It is essential amino acids. So in this way, this is the most important components of the amino acid for the body. To totally other 20 amino acids. Out of these 20, two more are the essential, two more are the non-essentials. The next topic is the amino acid pool. And in amino acid as the basic structural constituents of the naturally occurring proteins, they all consist of amino groups NS plus 3, a carboxylate group CO, a COO, a hydrogen atom, and a substituent group are called a side chain bounded to a central carbon atom, that is a C12 C14 atoms. There are the 20 standard amino acids as a structural formula. The amino acids in a amino acid pool, an adult has about 100 gram of free amino acids, which represent the amino acid pool of the body. The amino acid pool may be an oversimplification of the facts, since there is no single compartment, rather several compartments exist. Glutamate and glutamine together constitute about 50% and essential amino acid about 10% of the body pool. It is 100 gram. The concentration of intracellular amino acid is always higher than the extracellular amino acids. Amino acids enter the cells against concentration gradient by active transports. Means the amino acid pool is maintained by the source that contributes input and the metabolic pathway that utilize output. The amino acid as described below. As a specific stage to conducting or takes place in amino acid pools, as the three out of these two stages, the first stage is the sources of amino acid pool. This is sources of amino acid pool: turnover of the body protein, intake a direct dietary protein, and the synthesis of non-essential amino acid contribute to the body amino acid pool. So in this way, the first method is a protein turnover. In a protein turnover is the important things to the protein present in the body is an dynamic state. It is estimated that about 300 to 400 gram of protein per day is constantly degraded and synthesized, which represent body protein turnover. There is a wide variation in the turnover of individual proteins. For instance, the plasma proteins and digestive enzymes are rapidly degraded, their half lives being in hours or day. The structural proteins, example collagen, have long half lives, often months and years. So, in your protein turnover point of view, that is control of protein turnover. In control of protein turnover, the turnover of protein is influenced by many factors. A small polypeptide chain called ubiquitin molecular weight is 8,500 tags with the proteins and facilitates degradation certain protein with amino acid sequences proline, glutamine, serine, and threonine, etc. are the rapidly degraded in your particular view of the protein turnover. Next is the B, dietary proteins. In a dietary proteins, there is a regular loss of nitrogen from the body due to degradation of amino acids. In healthy adults, it is estimated that about 30 to 50 gram of protein is lost every day from the body. There is no storage form of the amino acids as the case of carbohydrates, glycogen, and lipids, triglycerols. The excess intake of amino acid is metabolized, oxidized to provide energy converted to glucose or fat. The amino groups are lost as urea and excreted. The protein consumption in developed countries is much higher than the RDA requirement of daily allowance. The daily protein intake by the adult in most countries is 40 to 100 gram. Protein is digested by proteolytic enzymes to amino acids, which are absorbed in the intestine and enter the body pool of amino acids. So in this way, this is a C, that is a synthesis of non-essential amino acids. In a synthesis of non-essential amino acid, 10 out of 20 naturally occurring amino acids can be synthesized by the body, which contributes to the amino acid pool. In amino acid pools, first is the sources of amino acid pool. Second is the in a sources of amino acid pool, there are the three subtopics. Small a is the protein turnover, small b is the dietary protein, and small c is the synthesis of non-essential amino acids. Next, 2 is the utilization of amino acid from body pool. In this steps, small a, that is the most of the body proteins 300 to 400 gram per day degraded as synthesized from the amino acid pool. This includes enzyme, hormones, immunoproteins, contractile proteins, etc. In your specifically to utilize to as your view to B, small b is the many important nitrogenous component which propyrines, purines, pyrimidines, etc. are produced from the amino acids. About 30 gram of protein is daily utilized for this purpose. In a C point, small c, many important nitrogenous compound which propyrines, purines, pyrimidines, etc. produced from the amino acid. About 30 gram of protein is daily utilized for this purpose. In C, generally about 10 to 15% of body energy requirements are met from the amino acids. And in D, The amino acids are converted to carbohydrates and fats. This become predominant when the protein consumption is in excess of the body requirements. So all kinds of the stages are required in the specific diagrams. To intimates is a figure 3.1, that is amino acid pool sources and utilization. So this is the first in your body proteins, uh, also 10 to 10 kg in adults. Protein breakdowns 350 to 400 gram per day. We also in a second body amino acid pools 100 grams. This also utilize dietary proteins 40 to 10 gram per day. Synthesis of non-essential amino acid variables to this is sources, and it util protein loss from body 30 to 15 gram per day, mostly as urea. Last is the urine. So this body amino acid pool 10 grams, also to pass protein synthesis 300 to 400 gram per day, also to synthesis of non-protein compounds 3 gram per day, creatine, propyrin, phospholipids, purines, pyrimidines, etc. Also as evolved by carbohydrates, fats, and third is the energy 10 to 50% of body's daily requirements. This is the utilization, this is the source. So this is a schematic representation and overview of the body pool of amino acids with the source and utilization is shown in figure 3.1. The amino acid pool of the body 100 gram is maintained through a simultaneously supply and utilization of amino acids. So this is a schematic representation of this. Dear friends, in your uh, protein metabolism, this topics will completed. Next topic of the chapters of units, general aspects of amino acid metabolism are discussed in the next lectures. Please, we also explain the briefly in a brief view as amino acid pools. That is next is a briefly to concept related already discuss amino acid pools. Amino acid pools as essential amino acid, non-essential amino acids. In a short view, the classification of amino acids, that is amino acids or I groups. There are 20 different types of the amino acids are takes place. The types of proteins, that is according to the environmental condition and general structural proteins can be roughly divided in three classes: fibrous protein, membrane proteins, and globular proteins. This is a specific category kinds of the proteins also used in different parts of the living organism body. The structural organization of the proteins, it is a primary structure of protein, first structures, secondary structure, second structure, tertiary structure is third structure, and quaternary structure is the fourth structure. This is a diagrams of this primary structure of protein, secondary structure of protein, tertiary structure of protein, and quaternary structure of. This is a detail continued by the description or theoretical point of view. Primary structure, secondary structure, tertiary structure, quaternary structure. This point of view is a linear sequence of amino acid in the polypeptide chains as the primary structure of the proteins. In secondary structure, alpha helix is the three-dimensional or arrangement of the amino acids with the polypeptide chain in a corkscrew shapes. Beta pleated sheets polypeptide chains also arranged side by this in a secondary structure of the protein. In tertiary structure, the overall three-dimensional shape of a polypeptide chains in these diagrams are indicated by the schematic representation. In quaternary structure, the combination of the two or more tertiary polypeptide that makes up the protein is a quaternary structure is the best example by the hemoglobin proteins. In your particular view, next topic of general aspects of the amino acid metabolism are discussed in next lectures. Dear friends, thank you. Stay home, stay safe.